Lars Ola Bygren was sitting in Uppsala, staring at church records from a village most maps don't bother naming, when he found something that broke the floor open beneath the entire field of genetics. Not a theory. Numbers. Flat, Swedish, indifferent to what they implied. The village was over Kalix in the far north, a place where winter comes in like a sentence, where famine years weren't metaphor, but arithmetic. He was tracking births, deaths, harvests, the ordinary ledger of survival. And then the pattern appeared, and it didn't fit anything he was supposed to find. The grandsons of men who had experienced famine during a specific window of childhood, a narrow band just before puberty, were living measurably, statistically longer. The granddaughters of women who had experienced abundance during that same window were dying earlier of cardiovascular disease. Not the men themselves, their grandchildren, a body not yet born, not yet conceived, carrying the metabolic memory of a winter its grandfather had endured in a village that history forgot. Bygren sat with those columns the way you sit with something that doesn't fit the room you thought you were in. What the data was saying, without drama, with the flat certainty of parish records, was that the lived experience of one body can rewrite the biological instructions carried by a body that doesn't yet exist. Not the genes, something riding on top of the genes, something that responds to scarcity, to stress, to the specific physiological and emotional conditions a person inhabits for long enough that the body begins treating them as permanent, as instructions. That is where epigenetics lives, not as a concept, but as a consequence. It entered science not through a theory, but through a column of numbers in Uppsala that refused to behave. Now here is where this stops being history and starts being something you need to sit with directly. If your biological baseline, your cellular architecture, your inflammatory thresholds, your nervous system's default state was partially configured by what your mother's body was running as its ambient signal during the months she carried you, then the story of your health did not begin with your first breath. It began in someone else's body, in a season you never lived through, in conditions you never chose. This is what the law of vibration means at the level of the cell, not as metaphor, as mechanism. The dominant frequency a body runs, its chronic stress load, its emotional weather, the signal its nervous system broadcasts as normal, leaves a biological residue. And that residue is heritable. It passes forward. It shapes what arrives, which immediately opens a second question. If it passes forward, does it also respond to what you do now? This is where biohacking stops being about optimization and starts being about something more fundamental. The interventions that research keeps returning to: breath regulation, cold exposure, deliberate nervous system reset. The structure practices that shift what signal your body is broadcasting a baseline are not wellness routines. They are attempts to update the instruction set, to write something new over what was written in conditions you didn't choose. The law of attraction, stripped of every layer of mysticism, points at exactly this. What you consistently run as your internal state determines what your biology encodes as normal. And what your biology encodes as normal determines what you're capable of sustaining, building, and passing forward. Bygren found the trail in a dead man's famine. The question this series is actually built around is what you do with the trail once you know it's there. The answer came from a spreadsheet, not from a lab with gleaming equipment, not from a clinical trial with enrolled participants who signed consent forms and showed up at scheduled intervals. From a spreadsheet rows and rows of health data belonging to identical twins, stretching back across decades, and a researcher sitting alone with that data who kept arriving at the same uncomfortable question. If the genes are identical, why aren't the outcomes? That question has been sitting in this series like an open wound. We raised it, we left it, and now the data has something to say. Not a partial answer, not a directional finding. An answer with 44,000 pairs behind it and 4 decades of tracking underneath that. The study came out through Science Alert, drawing on the Swedish Twin Registry, one of the most rigorous longitudinal data sets in modern epidemiology. The researchers weren't looking for something exotic. They were looking for patterns. Who got sick, who didn't, who lived longer, and whether the divergence between identical twins could be explained by the genes they shared. It couldn't. A consistent, measurable divergence between people with identical genetic starting points. The DNA matched. The trajectories didn't. And the gap was not small, not marginal, not the kind of thing you file under noise and move on. What the researchers began tracking instead were what the study called environmental exposures. But that term does a disservice to what was actually being measured. We hear environmental, and we think pollution, neighborhood, diet. Some of that was included. But the deeper layer pointed somewhere harder to look at, to what happened to these people before they had language to describe anything. The stress load carried by the mother during gestation. The nutritional availability during the first years of life. The hormonal climate of the household during early childhood. Cortisol levels in caregivers, the biochemical background noise of the environment a developing body was learning inside of. The body was being calibrated, set to a frequency, though the researchers didn't use that language. They used terms like epigenetic methylation patterns and inflammatory baseline and HPA axis sensitivity. Different vocabulary, same phenomenon. The system governing how your body responds to stress, how your immune system fires, how your cells age. That system was being configured during a period you have no memory of. You were not there for it consciously. Your body was there entirely. Now here's where I want to stop and not rush past this. Because there is something particular that happens when you actually let this land. You are 45, 50, maybe 60 years old. You have been told in various forms your whole adult life that the choices you make now determine the outcome. The supplement timing, the sleep hygiene, the way you eat, the way you move, the attitude you carry towards stress. And some of that is real. We are not throwing any of it out. But what the twin data is saying underneath all of that is something that takes a moment to absorb. The inflammation markers your doctor measured last spring have a history that precedes any decision you ever made. The way your immune system responds to stress. It learned that response during a time you cannot access. The cellular aging patterns showing up now in your blood work carry timestamps from decades you did not consciously live. This is not a metaphor. It is not a philosophical frame someone attached to the data after the fact. Sit with that for a second before we keep moving. Your body is not a blank slate that has been gradually marked by adult choices. It is the current expression of a biological document that started being written before your first breath. And the dread that comes with that, the specific, quiet dread of learning your body was shaped during a period you had no say in. That is an appropriate response. It doesn't need to be solved immediately. It needs to be felt because it changes what you are actually working with. The researchers tracked something called DNA methylation, chemical tags that attach to the genome and regulate which genes get expressed and which stay quiet. These tags accumulate over time. They are remarkably stable, and early life environments set the initial methylation architecture in ways that persist for decades. What the twin data showed was that twins who diverged in their health outcomes had diverged at the methylation level long before any clinical symptoms appeared. The disease was not a sudden event. It was the end point of a trajectory that began before the person had any knowledge they were on one. This is where the law of vibration becomes more than a useful phrase. Every biological system in the body operates at a frequency, a set point established early, running beneath conscious awareness, governing how you process threat, how you recover, how fast you age at the cellular level. This is not motivational language dressed up in science. The methylation patterns are the mechanism. The HPA axis calibration is the mechanism. The inflammatory baseline is the mechanism. What the metaphysical traditions called vibration, the biology is now calling epigenetic architecture. And the point both are making is the same. The frequency you are operating at right now was largely set by conditions that preceded your awareness of yourself. That does not make it fixed, but it means that optimizing at the surface, the supplements, the protocols, the mindset reframes, without addressing the deeper set point is like adjusting the thermostat without checking what the thermostat is actually wired to. 44,000 pairs. For decades, one of the longest-running human data sets ever assembled for this purpose. The film, not a snapshot, a film, shows that longevity is not written at 45. It was being written long before that. A person in their midlife today was born into a particular decade, a particular family's economic stress or stability, the hormonal environment of a mother who may have been calm or frightened, supported or exhausted. That context left marks, not only as psychological experience, that's a separate conversation, but as biological calibration. Factory settings, almost, except the factory was a human body navigating a human life under conditions it did not choose and could not yet name. The researcher in the spreadsheet was not asking why twins got different diseases. The question was why twins with identical genes ended up with different biological ages. And the answer pointed not to what they chose after 40, but to what their bodies absorbed before 5. The data is real. The sample removes ambiguity. And knowing this, actually knowing it rather than half knowing it, changes the question you need to be asking about your own body, which is exactly what the next part stays with. Not the broad finding, but the specific, what was actually being measured, what biological signatures are running in your body right now, and what they're telling the story of. That gets more precise from here. Thursday, November 29th, 1944. The city of Amsterdam has not had bread for 6 weeks. Not ration bread, not expensive bread, no brood. The Nazi blockade cut off Western Holland from food supplies in October, and by the time the first snow fell that year, families were burning their furniture for warmth and boiling tulip bulbs to extract something, anything, with caloric value. The official ration had dropped to 400 calories a day, a quarter of what the body needs to sustain basic function. The pregnant women of Holland ate bark soup and grass, and, in some documented cases, nothing at all for days. That winter is called the Hunger Winter. The Dutch hunger winter. Roughly 20,000 people died of starvation before the Allies broke through in May of 1945. The survivors went back to their lives. They rebuilt. They had children. Many of those children had children. And here is where the story takes a turn that nobody in 1945 could have anticipated. Those children, born to mothers who starved, grew up and were studied by epidemiologists decades later. Then their children were studied. What the researchers found was not what anyone expected. Bastian Hymans and colleagues, publishing in the Proceedings of the National Academy of Sciences in 2008, found that the children of women who were pregnant during the famine carried measurably different patterns on a specific gene called IGF-2, a gene involved in growth and metabolism. Different from siblings born before the famine. Different from siblings born after it. Still measurably different 50 years later. And different in ways that showed up in the body. Elevated rates of metabolic dysfunction, higher cardiovascular risk, altered glucose regulation. Not memory, biology. The cold did not do this. Fear in the abstract did not do this. The specific, sustained signal of scarcity reached into the developing biology of those children and turned certain switches in a particular direction. And then, in subsequent research tracking the grandchildren of those same women, traces of the same patterns appeared again. Not as strong, but present, measurable, real. Think of it this way. Your DNA is less a fixed sentence and more a vast room full of documents. Every page already written, nothing added, nothing removed. What changes is which documents the body pulls from the shelves, which files it opens every morning and reads carefully, which ones it seals and leaves in the back. The environment does not rewrite the archive. It controls access to it. It decides through small molecular flags attached to the genome, through methylation marks and histone modifications, what gets read loudly, what gets whispered, what gets silenced entirely. And what the Hunger Winter cohort revealed is that those access decisions can be passed forward, not through mutation, through the annotations. The flags stay on the documents. The next body opens the archive and finds the shelves already organized by a winter it never lived through. There's a feeling this produces when it arrives through the story rather than through a textbook. It is not quite dread. It is closer to the sensation of walking into a government archive, that specific chill, the dryness at the back of the throat, the smell of paper that has not been touched in decades, and finding a file with your name on it, and opening it, and recognizing the handwriting as someone else's. That is the vertigo the Dutch data delivers. Because what your blood sugar does under stress, how your cells allocate energy when resources feel thin, some portion of that operating logic was written in a winter you were not alive for, by a body responding, with complete biological sincerity, to a German blockade on the waterways of Holland. The Dutch cohort is not the only proof. Studies of children born during the 1959 to 1961 famine in China, the Great Leap Forward period when an estimated 15 to 45 million people died, found measurably altered metabolic and cardiovascular profiles in the generation born during and immediately after peak starvation. Published in Nature Communications in 2019. Effects tracking into adulthood. Parallel findings. Different continent, different political catastrophe, same mechanism. Except the Dutch data carries as something the Chinese data carries too. something that is easy to let slide past if you are reading quickly. The grandchildren. The second generation signal. The archive being organized not once, but twice by a winter neither generation experienced directly. Here the sensory chill in the archive sharpens into something personal, because you're holding documents you did not file. The women who survived the hunger winter were not unusual. They were not fragile or broken. They were ordinary people living through an extraordinary signal. And their bodies, because bodies are extraordinarily literal, took this signal seriously. Built a biology calibrated to a world trying to kill them, and handed some of that calibration forward, folded into the documentation like a letter tucked between pages. The mechanism is not historical. It is present tense. It is running right now in every body, reading the signal the environment is currently sending, deciding which files to pull and which to seal. And this is where the ground shifts from vertigo into something that has weight and use. Because if the body is this responsive, if a sustained environmental signal can reach into the deepest organizational logic of human biology and reorganize it, then the signal your body is receiving right now is not a minor variable. It is the question. Is what is being written into the archive at this moment, in real time, in your cells with your name on the file? This is the principle the law of vibration points at, not as metaphor, not as comfort, but as mechanism. The body is not a static object. It is a receiver, constantly reading the frequency of its environment and organizing itself in response. Sustained signals of scarcity produce one kind of biology. Sustained signals of safety, sufficiency, and possibility produce another. The Dutch mothers could not choose the winter, but you're not inside a blockade, which means the question of what signal you are generating, what the environment you are inhabiting internally and externally is telling your biology about the world, is one you have more authority over than the archive in your hand suggests. The letter was written by other hands. That is real, and it deserves to land as real before anything else is said about it. Several hands, across generations, each one writing under conditions they did not choose, leaving annotations your body still reads every morning when it decides how to allocate its resources. But the archive is not closed. The flags can move. The access decisions are not permanent. The biology that was organized around scarcity can, under the right sustained conditions, begin to reorganize around something else. That is not a promise. It is a mechanism. And the difference matters because one of those things you can actually work with. There was a study published a few years back out of McMaster University. Researchers followed adults into their 40s and 50s, people with stable jobs, stable homes, no ongoing crisis of any visible kind. And what they found was that the body was still braced. Cardiovascular reactivity, inflammatory markers, sleep architecture, all of it still running at settings that matched not the life these people were living, but the life their bodies had been told to expect before they were ever born. That finding landed in the research community with a kind of quiet force. It didn't make headlines the way a drug trial does. But if you've ever sat in a room where nothing is wrong and felt your shoulders refuse to drop, it will make sense to you immediately. Something in you has already lived this. Here's what the biology actually shows. The marks left on your DNA by experience, by stress, by hunger, by the particular texture of silence inside a household. Don't ask for your consent. Methyl groups attach, to cytosine bases. Histones tighten or loosen around strands of genetic code. No morning you woke up and agreed to it. The body simply recorded what was in the air, the way a wall records water damage, even after the leak has been fixed for 30 years. Psychologist Bruce Ellis called it anticipatory calibration. The developing child is not passively receiving conditions, it's actively reading them. Cortisol levels in the mother's bloodstream, inflammatory markers, the presence or absence of hormones tied to safety and available resources. The fetal body is processing all of this not as background noise, but as a forecast. It is asking a question about what kind of world is waiting, and then it builds itself accordingly. If the signal says scarcity, the metabolic systems optimize for storage. If the signal says threat, the stress response threshold is set low, fast to fire, slow to come back down. If the signal says unpredictability, the nervous system learns to stay partially on, the way a dog in a loud house never quite sleeps all the way through, ears still moving even in the quiet. None of this is the mother's fault. Ellis was careful to say that. The researchers at the University of New Mexico, who extended his work into adult health outcomes, said it too. The calibration isn't a punishment. It isn't a verdict. It is the body doing exactly what bodies are built to do. Preparing the next generation for the world it has been given reason to expect. The hard part is this. The world it was told to expect and the world you're standing in right now may have almost nothing in common. The child whose body calibrated for scarcity might be sitting in a house with a full refrigerator, a steady income, no real immediate threat. And the systems that were set for otherwise are still running, still scanning for the shortage, still treating every surplus as temporary, still holding the metabolic dial at a setting that made sense in a context that closed decades ago. The McMaster study tracked this directly. Adults whose early developmental environments showed hallmarks of high-threat calibration, assessed through maternal stress records, household income, neighborhood instability, carried measurably different baselines into their 40s and 50s even after 20 years of objectively stable adult life. The body did not update. The forecast was issued before the person drew a first breath, and in many cases the biological systems never got word that the forecast was wrong. That's the thing that hits. Because what it means is that a real portion of what it feels like to be in your body right now, the vigilance that arrives before any conscious thought, the way certain silences feel faintly loaded, the specific weight of a Sunday afternoon that carries unease for no reason you can name. That may not be a response to your life as it actually is. It may be a response to a life that was anticipated for you before you were born. You didn't choose the calibration. You didn't sign the blueprint. And yet here it is, still running this morning in the background of everything. Daniel Lenda called it a mismatch between the prepared organism and the actual environment. The word mismatch is too clean for what it feels like. What it means on the ground is that you may be extraordinarily well equipped for a world that no longer exists and only partially calibrated for the one you're actually in. And here's where it stops being just biology. The law of vibration tells us that everything is in motion, including the internal state you are broadcasting in any given moment. Not the story you tell about yourself. The actual signal your nervous system is running, your dominant emotional frequency. And if the frequency you're running was set in a womb, calibrated for a scarcity that never came or a threat that long since passed, then the world you're calling toward yourself is being filtered through a lens that was ground before you were conscious enough to question it. This is not fatalism. It is the opposite, because the calibration is not a life sentence. It is a setting, and settings can be changed, not through willpower, not through argument with your own nervous system, but through the sustained practice of introducing new signal, new dominant frequency, new evidence delivered not as an idea, but as a felt experience, repeated enough times that the body begins to update its forecast. The organism learns what the environment keeps proving. That was the original mechanism. It still is, which means the same system that locked the calibration and can be the system that revises it, if you understand what you're actually working with. You are not broken. You are not behind. You're running a setting that was written for someone else's world, and you're more capable of changing that than anyone told you to expect. The question that hung open in the last chapter, what exactly the organism of a child reads in the womb to calibrate its systems to the world, deserves a precise answer. Not a metaphor, a mechanism. Here it is. The fetus does not have eyes trained on the world outside. It has no access to news, no cortex yet fully wired for language, no framework for understanding whether the environment beyond the uterine wall is safe or dangerous. What it has is chemistry. Specifically, it has receptors, tiny docking stations built into the developing hippocampus. They're designed to read the hormonal signature of the body it lives inside. And the hormone those receptors are reading most consistently, most consequentially, is cortisol. Cortisol crosses the placenta. Not all of it. The placenta produces an enzyme, 11-beta hydroxysteroid dehydrogenase type 2, if you want the exact name, that acts as a partial filter, converting some of the maternal cortisol into an inactive form before it reaches the fetus. But the filter is not a wall. Under chronic, sustained stress, not acute, not occasional, but the kind of stress that runs for weeks or months, the kind that wires itself into a person's resting baseline, the filter gets overwhelmed. The enzyme cannot keep pace. And the cortisol that gets through lands on receptors in a hippocampus that is still deciding how many of those receptors to build, how sensitive to make them, how the whole stress response architecture will be configured for the rest of that organism's life. Researchers at King's College London have documented this. So have teams at the University of Auckland. The finding, replicated across different populations in different decades, is not subtle. Elevated maternal cortisol during gestation, chronic, not occasional, alters glucocorticoid receptor expression in the developing hippocampus. The system does not break. It calibrates. It reads the hormonal environment as information about the world and sets its sensitivity accordingly. If the chemical signal says danger is frequent and sustained, the system that gets built is one primed for danger. More reactive, faster to trigger, slower to return to baseline. The developing brain, in other words, is not receiving trauma. It is receiving a weather report, and it is building a code accordingly. There is something in this that is almost elegant if you can hold it without flinching. Evolution did not design this as a flaw. A feed is born into a genuinely high-threat environment. A drought, a war, a famine that is pre-calibrated for high alert has a survival advantage over one that arrives expecting safety that does not exist. The system is adaptive. The problem is not the design. The problem is that the signal does not always correspond to the actual world the child will inhabit. The mother's sustained cortisol may have been generated not by genuine physical danger, but by a bad marriage, a failing business, a country in the middle of a recession, an apartment too small, a body that never stopped bracing. The fetus reads the chemistry. It cannot read the context. The result is a nervous system calibrated to a world that, in many cases, no longer exists by the time the child is 30 or 45 or 60. This is worth sitting with. Not with blame. Blame would actually be simpler, but with something more disorienting than blame. A kind of recognition that the architecture you have been working with your whole life, the reactivity you have been managing, the baseline tension you have been treating as personality, may have been installed before your first breath as an adaptive response to someone else's sustained internal state. That reframe is not a comfort, but it is a different kind of problem. And different kinds of problems open to different kinds of work. Because what changes when you name a mechanism is the distance between you and it. Not that the mechanism stops operating. It does not. Not immediately, not by naming alone, but that you stop mistaking it for identity. The hippocampus that was calibrated in utero is real. The receptor density is real. The hair trigger reactivity that certain people with that calibration carry into their 50s and 60s is real. None of this is invented. None of this is weakness. But it is not the same as, this is who I am. What it is, technically, is, this is what the environment of another person's internal state built inside me. The inverse is also documented. The same King's College research, extended into follow-up studies, found that maternal emotional environments characterized by sustained low-threat activation. The term in the literature is something close to calm arousal, a state of engaged but non-threatened presence, produced measurably different fetal cortisol exposure and, correspondingly, different receptor calibration outcomes. The hippocampus still got built. The stress response system still got configured. But the configuration reflected a different weather report. Less threat in the chemistry, less hair trigger in the resulting architecture. The sustained internal state of one person became the biological environment of another. Measured, documented, replicated. What this means is not that you are defined by what happened inside a womb. Epigenetic expression is not a fixed sentence. The hippocampus retains plasticity into late adulthood. This is well established, and a later chapter goes into the specific mechanisms. What this means, right now, is something more foundational than any of that. The state internal to a person is not private. It is not contained inside the skin of the person experiencing it. It radiates into chemistry. It crosses barriers we assumed were sealed. It writes itself into the tissue of whoever is living in proximity to it, in sustained, intimate contact with it. The fetus is the most extreme example because the exposure is total and the receiver is the most plastic organism imaginable. But the principle does not disappear at birth. If the sustained emotional state of another person became your biological environment before you had a name, before you had a thought, then the question that follows is not abstract. It is, what is your sustained internal state becoming for someone else? Right now, today, in the bodies and nervous systems of whoever is living in close proximity to yours. Not just children, not just the people you're raising, but anyone whose chemistry your chemistry is touching with any regularity or depth. What environment are you building in them? That question does not resolve here. It opens. And the part of the answer that most people do not expect, the part that determines what any deliberate change actually requires, is that it does not begin with behavior. You can change what you do without changing what you are generating beneath what you do. The people living in proximity to you are not reading your behavior the way a stranger reads it. They are reading your chemistry. They are living inside your weather, which means the intervention, if there is one, has to reach below the level where most interventions aim. That is where the next chapter begins. The answer had been sitting in the data for years before anyone thought to look at it that way. The open loop from earlier, whether the emotional state of a mother during pregnancy actually reshapes fetal biology, and whether that mechanism persists beyond the womb, lands here. Not with a dramatic reveal, with something quieter, more uncomfortable. The answer is yes, and yes again. The mechanism does not stop at birth. It has never stopped. What the research shows when you follow it past the headlines is that you are not simply the product of what happened before you arrived. You are still being written. That changes things. Michael Meany was not looking for a philosophy when he started his work at McGill University. He was looking at rats, mother rats, specifically watching them in the first days after birth, the way they moved over their pups, tongue and weight and warmth, the smell of milk and cedar shavings in a laboratory that had no windows, the scratch of a pen on a clipboard, the hum of ventilation. A researcher counting something that most of his field would have called irrelevant to hard science. The frequency of licking, the quality of contact, small things, biological things. He found they were not irrelevant. What Meany's team found, and what is held up through replication in ways that matter, was this: the pups that received high levels of that early contact grew into animals with a fundamentally different stress response profile. Not different in mood, different in tissue. The methylation pattern on a specific gene, the one governing glucocorticoid receptors, which calibrate how a body responds to threat, was measurably altered by the quality of care received in the first week of life. More contact, less methylation. Less methylation, more flexibility. Less care, heavier marks on the gene, a stress response system that stayed reactive, harder to regulate, years later. This was not temporary. It showed up in the behavior. It showed up when those pups became mothers themselves and, without instruction, groomed their own offspring at the same frequency they had been groomed, not passing it through the DNA sequence, but through the act of care itself. The pattern moving forward not as code, but as conduct. There are epigenetic marks that appear to be deeply stable, established early, maintained through cell division, functionally durable within a human lifespan under ordinary circumstances. If you grew up in an environment of chronic unpredictability, of emotional dysregulation, of care that came intermittently or not at all, some portion of that history lives in your gene expression. Not as memory, as architecture. The weight of that is real, and it has a texture to it. The way cold metal has a texture when you press your palm flat against it and hold it there. It doesn't yield immediately. It doesn't pretend to be warm. So, some of it may not reverse. That is true. And here is what is also true, and what Meany's research actually shows when you read past the first paragraph. The process never stopped. The same mechanism that wrote those early marks, gene expression reading environmental signal, chromatin shifting in response to chemistry, did not close at birth. It did not end at 2 years old, or at 7, or at 45. The methylation machinery is still operating. The question of what it is currently reading is not a settled question. It is the question you are living inside right now, whether you know it or not. You are an environment, not metaphorically, biologically. The cells that carry your epigenetic history exist inside a chemical environment that you generate continuously through emotional state, through breathing patterns, through the sustained quality of nervous system activation hour by hour. That environment signals. It signals in the same fundamental language that maternal grooming signaled to those rat pups, through chemistry, through sustained conditions surrounding the genome. The quality of the air in the room, the weight behind the eyes, the way a jaw either holds or releases across the length of an afternoon. The body is reading all of it. It has always been reading all of it. The early marks were written by an environment you did not choose. The current environment is one you are generating, not through willpower, not through awareness alone, through the aggregate of how you move through your days, what you attend to, what registers as threat, what you allow to pass through without taking up permanent residence. This connects directly to something the research in this series keeps circling back to, the idea that coherent internal states, states in which emotion and physiology are synchronized rather than fractured, produce measurably different biological signals than states of chronic low-level alarm. This is not a metaphor for feeling better. It is a description of what the body broadcasts to itself. The law of vibration, applied at the cellular level, is not about optimism. It is about signal quality. What frequency is your internal environment running at, right now, continuously beneath the level of your attention? Because that frequency is the grooming. That is the lick and the warmth and the sustained presence. That is the condition in which your own gene expression is occurring. And the biology does not distinguish between a signal that arrives from outside you and one that you are generating from within. What you are, right now, is in part a record of every environment you have passed through. And you're also the current environment for every system in your body that is still reading the signal. The mechanism is not mystical. The methyl groups either sit on the gene or they do not. The receptor is either expressed or it is suppressed. The chemistry is real and it responds to real conditions. What Meany's work demonstrated, what became the foundation for an entire subfield, was that the quality of sustained emotional environment produces effects at the level of the genome that are both measurable and lasting. Not supplements, not clinical procedures, the quality of the conditions, the sustained signal of safety or its absence. You're that signal. You are broadcasting it right now. In the temperature of your attention, in the texture of how you're holding this moment, in the low persistent hum of whatever your nervous system is treating as normal. The process that began before your birth has not paused. It is running in the cold clarity of a room where the window is open just enough, where the air has an edge to it that makes things feel more precise, more awake, more like something that can still be shaped. What moves it? What specifically and measurably shifts the signal is where this goes next. And it is not a small answer. There is a particular kind of mourning that people in their 40s know without needing it described. The alarm has gone off. The body is technically awake. But before a single decision has been made, before coffee, before the phone, before the first word spoken to anyone, something has already set the tone. A low-level hum of readiness for difficulty. A slight contraction in the chest that is not pain and is not fear, but is somewhere in that neighborhood. The day has not asked anything of them yet, and the body is already bracing. Most people who experience this have learned to move through it. They are functional. By most visible measures, they are fine. They would not describe themselves as suffering. They would describe themselves as tired, maybe realistic, a little worn. The kind of worn that accumulates without a single identifiable cause. The way a wooden step wears not from any one footfall, but from years of ordinary use. What they would not think to say, because no one told them this was a thing to say, is that this is a biological condition, not a mood, not a character trait, not evidence of weakness or insufficient gratitude or failure to meditate enough. A biological condition with measurable downstream effects that has been producing specific instructions in their cells every morning for years. That is not a metaphor for how bad stress feels. It is a description of a mechanism that researchers can now trace with considerable precision. The field that has done the most rigorous work here is not wellness. It is not self-help. It is immunogenomics, specifically the branch of it concerned with how psychosocial experience translates into changes in gene expression. Stephen Cole at the UCLA School of Medicine spent the better part of 2 decades building the methodology to track this. And what his team found, replicated across studies with different populations and different methodologies, is the following: a sustained sense of threat, not a crisis, not a diagnosis, but the ordinary low-grade vigilance that becomes a person's default setting, produces reliable, measurable shifts in which genes are turned on and which are suppressed. The genes that increase activity under chronic threat conditions are predominantly associated with inflammatory response. The genes that decrease activity are predominantly associated with antiviral defense and cellular repair. The body, reading the signals arriving from the nervous system day after day, makes a resource allocation decision. It prepares for the threat it perceives as most likely. And if the threat signal is sustained, not because anything dramatic is happening, but because the nervous system has learned to hold a particular posture toward the future, the body holds its preparation posture indefinitely, at significant biological cost. Inflammation is in part that posture. It is the body prepositioning its resources. And chronic inflammation, the kind that does not resolve because the perceived threat never quite resolves, is one of the most consistent markers of accelerated biological aging across virtually every tissue system that researchers have thought to examine. Meanwhile, at the Salk Institute, a different thread of research was pulling on a question that seemed, at first, entirely separate. In 2013, a team led by Juan Carlos Pijua Belmonte began investigating whether the epigenetic programming of cells, the layered instructions written on top of the DNA sequence that tell a liver cell to behave as a liver cell and not as something else, could be partially walked back. The conceptual foundation came from the earlier Nobel Prize-winning work of Shinya Yamanaka, who had demonstrated that you could take a fully mature adult cell and, by introducing 4 specific transcription factors, return it to a pluripotent state. A cell that had, in a sense, been reminded that it contained more possibilities than it had been expressing. Belmonte's team was not trying to erase cellular identity. They were asking a more precise question. Could those same factors be used partially, briefly, cyclically, to reset some of the epigenetic aging markers without destabilizing what the cell fundamentally was? The mouse model results were striking. Subsequent research published in Nature in 2022 drew attention from researchers working in longevity science, aging biology, and fields adjacent to both. The principle it established is foundational in a way that is still working its way into mainstream conversation. The epigenome ages, and the epigenome can, under specific conditions, be partially reset. It is not a fixed record of what happened to you. It is a dynamic document still being written. Here is where the two threads meet, and the meeting is not subtle. Inflammatory cytokines do not appear from nowhere. They are produced by biological systems that are responding to conditions. And those conditions include, with documented specificity, the sustained emotional state of the person carrying the biology, the morning chest contraction, the habitual bracing, the years of holding a particular posture toward the world. These are not separate from the cellular environment that the Salk research was examining. They are how that cellular environment gets made. The cold comes off old stone buildings in winter. Not a dramatic cold, not a wall of it, but the particular seeping chill that finds you before you have consciously registered being outside. You do not feel it arrive. You feel it when it is already in your coat. That is how the sustained emotional field works in the body. Not as a single event with a timestamp. As a temperature, a prevailing condition, the background against which everything else occurs. And the epigenome is reading that temperature constantly, issuing instructions accordingly. The people who are going to push back on this are the ones who have already learned correctly to be suspicious of the version of this story that slides into blame. The version that implies that if you are sick, you chose it. That if your biology is compromised, you failed to think positively enough. That the marks left by a childhood lived inside other people's fear and silence were actually your responsibility to simply decide your way out of. That is not what the research says, and it is worth being clear about that with the same precision the research requires. The marks were real. The early epigenetic writing happened in an environment you did not select, could not have altered, and were in no position to resist. The body was doing exactly what a body is supposed to do, reading the conditions of its environment and building itself accordingly. That is not a failure. That is biology functioning as designed. What the research also says in the same papers and the same data sets is that the writing did not stop. The mechanism that received those early instructions is still active. It is still responsive, which means it is still responsive to conditions, including conditions that are now, for the first time, within your actual reach. The specific man who sits with a glass of water in a dim room at 6 in the morning, before anyone else in the house is awake, already running the calculation of what the day is about to ask of him. He is not thinking about cytokines. He is not thinking about epigenetic aging markers. He is just sitting with the familiar weight of the day before it has officially started. He knows this feeling. He has known it for years. He has assumed, the way people assume things that have always been true about them, that this is simply what mornings are. What no one has told him is that this assumption is not innocent. Not because he is doing something wrong, but because the assumption forecloses a question that the science has now made answerable. The question is not whether the habitual internal state he carries influences his biology. That is answered. The question is what specifically runs the signal from the state to the cell. What is the actual pathway, the traceable mechanism, that carries something as apparently non-physical as a habitual orientation toward difficulty and converts it into a biological instruction his genome receives and acts on? That mechanism exists. It has a name. It operates through systems that are not mysterious, not metaphysical, and not outside the range of what a person can learn to work with deliberately. And understanding it changes what is available in the hour before the house wakes up. The body you are living in right now is not a static object. It is a process, a continuous, unfolding biological event. And if you've been carrying something lately that you cannot quite name, a kind of background hum, a low-grade tension that sits just beneath the surface of ordinary days, there's a reason it feels like it lives in you rather than around you. There is a reason it does not leave when the circumstances that caused it change. The body does not just register that state. It learns it. It gets efficient at producing it. And once it gets efficient, it runs the pattern with very little input required. That is not a character flaw. That is a nervous system doing exactly what nervous systems do. I want to talk about a researcher named Richard Davidson who has been working out of a laboratory at the University of Wisconsin since the early 1990s. Davidson spent decades doing something most of his colleagues considered too difficult to be useful, measuring how sustained emotional states, not momentary feelings, not survey responses, but the baseline register a person inhabits across months and years, alter the physical structure and function of the brain and the immune system at the same time. His lab was not working from a wellness framework. He was using electrode data, brain imaging, and blood draws simultaneously over extended periods. And what he found has now been replicated across more than 30 years of research. People who sustain what Davidson called left prefrontal dominance, a baseline orientation toward engagement, approach, interest in what is ahead rather than vigilance toward what might go wrong, show measurably different immune responses than people who sustain baseline tilts the other direction, toward withdrawal, toward chronic threat anticipation. The kind of chronic anticipation that does not feel dramatic from the inside because it has been there so long it just feels like being awake. In one of Davidson's landmark studies, conducted in collaboration with John Kabat-Zinn and a group of employees at a biotech company in Madison, participants who went through an 8-week program aimed at shifting their sustained emotional baseline showed not only changes in prefrontal activity, but statistically significant increases in antibody response after flu vaccination compared to people in the control group. The emotional shift that appeared in the brain imaging also appeared in the bloodstream. The same week, measured independently. That is not a study about feeling better. That is a study about what your emotional baseline is physically constructing in the tissue. There is a biomarker worth knowing about here because it makes this concrete in a way that is hard to dismiss. Heart rate variability, the interval variation between consecutive heartbeats, is one of the most reliable non-invasive measures of how well the autonomic nervous system is regulating itself. And it tracks with emotional baseline more closely than almost any other accessible marker. A person who spends years in a state of low-grade ambient unease, not acute crisis, just a persistent background hum, that a lot of people over 45 carry, as though it were simply the texture of being responsible, shows a systematically suppressed heart rate variability. And suppressed HRV correlates with elevated inflammatory markers, accelerated epigenetic aging, and reduced prefrontal regulation, not as isolated readings, as patterns that compound across time. A study published in 2019 in the journal Neuroendocrinology followed middle-aged and older adults over 5 years and found that individuals with chronically low positive effect showed significantly higher circulating levels of interleukin-6 and C-reactive protein than those who maintained stable, moderate to high positive effect baselines. After controlling for diet, exercise, smoking status, and BMI, the emotional baseline still carried independent predictive power over the inflammatory picture. The state you inhabit is producing chemistry consistently, whether or not you are paying attention to it. Davidson has a phrase he uses carefully. He says that emotional states are not things that happen to the brain. They are things the brain learns to produce. And once the brain has been running a pattern long enough, it runs that pattern efficiently, automatically, on very little fuel. The body becomes a very good factory for whatever state it has been running the longest. Sit with that for a moment. If the state has been chronic vigilance, scanning for what might go wrong, bracing before the difficulty arrives, anticipating the loss before it happens, then the body has been manufacturing the full biochemical output of that state at scale for years. Not because something is broken, because the brain learned, because it was trying to protect you. And it got very good at the job. What Davidson's data also shows, and this part gets less attention in popular coverage, is that the relationship runs in both directions. The sustained emotional state does not only produce a biological signature, it shapes what the brain registers as worth noticing. People with chronic right prefrontal dominance, the withdrawal and threat pattern, show heightened attentional bias toward negative information. In the same stream of data that someone with a different baseline moves through without flagging, these individuals are more likely to notice the threatening signal, hold it longer, and encode it more deeply in memory. Which means the organism is not receiving the world neutrally, is filtering it. The attentional architecture built by sustained emotional state determines what gets through, what gets weighted, what gets remembered, and what kinds of signals, openings, and possibilities the person consistently notices versus consistently misses. This is the mechanism behind something people have called the law of vibration and the law of attraction, not as borrowed spiritual language, but as a description of what Davidson's controlled research has been measuring for 3 decades. A sustained emotional state is a sustained biological frequency. It produces consistent outputs in the tissue, in the immune profile, in gene expression, and the heartbeat interval. And it builds a perceptual filter that determines what the person actually receives from the environment day after day. You're not missing opportunities because the world is withholding them. You're often missing them because the filter built by years of vigilance was optimized for something else entirely. Outside, somewhere, wind moves through dried leaves with the sound of very light rain. Something the inner ear catches before the mind has a name for it. Something real that almost goes unregistered. That is what this is about. Not the state you perform when someone is watching. The state that runs the factory when no one is. The one that has been quietly shaping the tissue, the attention, the daily experience of what feels possible and what feels out of reach. And here is the thing that Davidson's data makes undeniable. The relationship runs both directions. If the sustained state shapes the biology, then shifting the sustained state shifts the biology. This is not wishful thinking. It is the same mechanism operating in the other direction. The factory can be retooled. It has been retooled in controlled studies with measured outcomes in peer-reviewed journals. You're not too far along. The body is not a fixed object. It is a process, and processes can change direction. That is not a small thing to know. There was a story out of the Netherlands in 2023 that most people didn't stop on. A research team at Maastricht University published a follow-up analysis of the Dutch hunger winter cohort. The children born to women who were starving during the Nazi blockade of 1944. These people were in their late 70s now, and the researchers were still finding it. Methylation patterns on specific genes set in the womb 80 years ago, still influencing inflammatory response, metabolic function, cardiovascular risk. The original signal still running. Most people who encountered that story filed it somewhere between interesting and depressing and moved on. If you're over 45, I don't think you moved on that fast. Because that story isn't really about the Netherlands. It's about the question you've probably been sitting with longer than you'd admit. Whether what's already happened to you, the decades of it, the accumulated weight of it, whether that's something you're steering now or something that's steering you. Whether the document was written before you had any say in it. That is not an abstract question when your body is giving you daily evidence. So before anything else gets introduced here, that deserves to be named plainly. Not reframed, not rushed toward a solution. You're carrying something real. The research confirms the weight is real. And anyone who skips past that to hand you a protocol hasn't actually understood what they're asking you to believe. What happened in the weeks after that Maastricht story ran was smaller and quieter. A cardiologist in Rotterdam, not part of the original team, started getting emails from people in their 50s and 60s who had read it, not asking about medication, asking whether there was anything the body could still do, whether the ledger was closed. He wrote a short piece for a Dutch medical newsletter, never translated, never picked up widely, saying something that has stayed with me. He wrote that epigenetics had spent 20 years establishing the weight of the past, and almost no time explaining that the same mechanism that records old signals can receive new ones. That is the thing that didn't make it into most of the coverage. The genome is not an archive. It is a live document responding to the internal environment of the body right now, this hour, with the same basic logic it used in the womb. Genes don't change. Gene expression does. And what drives expression is the sustained chemical and electrical climate inside the body. Not single events, not acute spikes, but the signal that runs continuously underneath everything else. Here is where something specific enters. The heart doesn't beat metronomically. It never has. Even in a healthy, resting body, the intervals between beats vary constantly, slightly closer, slightly further apart, in a continuous oscillation. That variation has a pattern, and that pattern shifts depending on what is running in the interior of the person, under sustained low-grade stress. The kind that has been present long enough that most people over 45 have stopped registering it as stress. The pattern becomes jagged, irregular. The intervals spike and drop without rhythm. and the signal traveling from heart to brain through the vagus nerve is essentially noise. The body reads that noise and responds in kind. Immune system primed, endocrine system on alert, the whole biological architecture in a half-emergency that never fully resolves. Not a crisis, the residue of one. The way a smell from a specific corridor, disinfectant over something older, can live in the body for years after the building is gone. Not sharp, just always there, under a different internal condition. Not happiness, not forced calm, something more precise than either. Low threat, clear attention, the specific quality of presence that accompanies genuine focus or care. The pattern shifts. The intervals organize into a smooth wave. The signal changes character. What arrives at the brain is no longer noise. This was documented. The HeartMath Research Center built a measurement infrastructure around it across the 90s and early 2000s. That work was replicated, extended. Then, in 2016, a team published findings in the American Journal of Health Promotion looking at something more specific. Whether this shift in cardiac pattern produced measurable changes in epigenetic markers. They were tracking DHA to cortisol ratios, immunoglobulin A levels, biological signatures of the sustained internal environment, not mood reports, not self-assessment. 6 weeks of practice, statistically significant shifts in the markers. The genome had received a different signal, and the signal had come from inside the body of the person practicing. Stanford Department of Psychiatry contributed to adjacent work showing that sustained generation of these states reduced inflammatory gene expression, not in animals, in adults whose epigenomes have been accumulating decades of environmental input. People not unlike the ones emailing the cardiologist in Rotterdam. People have been running on a jagged signal for 20 years and wanted to know whether that was permanent. It isn't permanent. That's what the data says. Not as reassurance. Reassurance is just noise with a friendly tone, but as a mechanically documented fact. The epigenome is a ledger still being written, and the instrument doing the writing right now is the sustained internal environment you are generating. The heart's rhythm is readable. The pattern it produces is trainable. The markers respond. The timeline is documented. What's harder to say, and this is where honesty requires slowing down rather than accelerating, is that knowing this does not produce it. The gap between understanding the mechanism and actually changing the signal is not closed by information. The body needs something more specific than knowledge. It needs a practice that is consistent enough and precise enough in its physiological signature to generate the state rather than just describe it. Not breathing exercises from an article. Something with a measurement protocol. Something with a documented timeline for when the markers begin to shift. That practice exists. A person can learn it without a clinic. It fits inside a day that is already full. And the reason to trust that isn't hope, it's that the measurement exists. You can watch the jagged line on a screen in real time, smooth into a wave. You can see the body translating a change in internal state into a change in the signal the genome is reading. That's not a small thing. After everything the Dutch cohort data implied about what's already been written, that is not a small thing at all. The answer was always there, tucked inside the question itself. Earlier in this video, we asked whether cardiac coherence changes epigenetic markers only for the person practicing it, whether the signal stays contained. Whether the work you do on your own nervous system is, in the end, a private matter. It isn't. Here is what the data says, and it says it plainly. Emilio Ferrer, a psychologist at UC Davis, spent years running a deceptively simple study. He recruited couples, not athletes, not meditators, not people who had signed up for anything unusual, just long-term couples living ordinary lives. He strapped instruments to their chests. He measured heart rate variability over time. What he found didn't make the evening news, but it sits quietly in the literature like something that was always true and finally got measured. The hearts of people who live closely together begin to move together, not metaphorically, in milliseconds, in waveform. The variability of one person's cardiac rhythm begins to entrain with the variability of the others. You inhale, they adjust. You regulate, they follow. The synchronization is not conscious. It is not chosen. It happens the way humidity enters a room, not through the door, through the walls. Ferrer's work was not about love in any soft sense. It was about physiology, about the body as a transmitter operating continuously, broadcasting into nearby space, and those nearby bodies receiving and beginning to mirror what arrives. Outside in the early morning, the grass still holds the cold from the night before. You feel it through your soles before you've even looked down, before you've registered that the temperature changed. The mechanism Ferrer documented works on a similar logic. It sits inside the same basic neuroscience that governs why you yawn when someone near you yawns, why your mood shifts in a crowded room before you understand why the first responder who stays composed pulls the injured person's nervous system toward composure. Polyvagal theory has a name for some of this. Ferrer's work puts numbers on it. Now hold that against what we have been building across this entire video. The research on prenatal epigenetics, the Dutch hunger winter cohorts, the studies on maternal stress and methylation patterns in offspring, established something that should have landed harder than it did. The sustained emotional state of another person operating in a body physically near or biologically connected to yours leaves measurable marks on your biology, not as a metaphor for influence, as a literal mechanism. Gene expression, stress response architecture, metabolic thresholds. These were shaped in part by someone else's internal weather. You receive that. You're still receiving. This is the chapter where the video becomes a mirror. Not a flattering one, not a harsh one, just accurate. If the sustained emotional state of your mother during those 9 months contributed to how your stress response was built, if her cortisol patterns, her vagal tone, her periods of fear or calm became architectural instructions for your nervous system, then the sustained emotional state you are carrying right now is doing something too. Not to a fetus, not to some distant biological outcome decades away, to the tissue you are living in today, to the people in the same room, to the person sleeping 2 feet away from you who is, without either of you discussing it, running a continuous low-level entrainment of your cardiac rhythms every single night, the same way an old wooden floor teaches you which boards to step over, gradually, without a single decision, until the knowledge is just in your body. The epigenome is not a vault that got locked at birth. The research on this point is no longer preliminary. Studies from the Salk Institute and work published in Nature Reviews Genetics in recent years have reinforced what the field has been converging on for over a decade. The epigenome remains responsive across the lifespan. Methylation patterns shift. Histone modifications respond to chronic input, not dramatic input, chronic input, not the emergency, the baseline, the emotional state you return to after the emergency passes. The hum you carry through ordinary Tuesday afternoons, in the car, in the silence after dinner, in October kitchen light that falls flatter and longer and cooler across the table while you sit in it without noticing it all. That is the signal your cells are reading. That is what gets written. None of this is an invitation toward guilt. A sustained guilt state produces its own chronic activation signature, its own methylation input, its own transmission to the people around you. So as a strictly biological matter, guilt here is not just unpleasant, it is counterproductive. If you walk away from this chapter feeling that you have failed the people near you by carrying stress, you have misread what is being said. What is actually being said is something closer to operational responsibility, not the punitive kind, the kind that belongs to someone who understands they are not sealed. The nervous system is permeable. The signal you are running, the one that started before you were born, the one you have been carrying and modifying and sometimes reinforcing and sometimes softening across your whole adult life. That signal is going somewhere. It is reaching the person across the table. It is reaching the body next to yours in the dark. Farah measured it. Others have replicated it. The data is there. This is also where the physics of the situation becomes impossible to sidestep. The law of vibration is not a piece of vocabulary invented by self-help culture. It is a description of what matter does. Everything oscillates. Every system broadcasting a frequency will, under the right conditions, begin to pull nearby systems into resonance with it. Farah's instruments documented that principle operating inside human cardiac tissue. The epigenetic research documented it operating across generations. What the law of attraction adds is directionality. The dominant internal state you sustain is not merely coloring your experience of incoming events. It is functioning as a selection filter for which events reach you at all, and as a transmission shaping which states the people nearest to you are being pulled toward. These are not separate claims. They are descriptions of the same underlying dynamic at different scales. There's a version of this information that settles on you like weight. Another person's biology is downstream of your state. The people closest to you are in training to something you carry. The feeling of that, if it arrives, is appropriate. This is not trivial, and the voice that tells you it is fine to dismiss, it is not your most honest one. But there is another version, and it is the more accurate one. If you have spent any part of your life waiting for some external condition to change before you could settle, some financial threshold, some political resolution, some relationship repair, some health result, the research we have been walking through today suggests the direction of causality has been inverted. The internal state is not the reward at the end of the work. It is the instrument doing the work. It is the signal being transmitted. The ordinary, private, invisible labor of bringing your own nervous system toward coherence, away from chronic emergency activation, back toward a baseline that your cells can actually build from. That labor has a radius. It extends past your skin. It reaches the bodies near you. It shows up in data that Farah collected in a university lab using instruments that had nothing to do with spirituality and everything to do with measurement. You're not a closed system. You never were. The question this raises is not whether you're transmitting. You are. The question is what? And unlike every question this video has asked before it, this one doesn't resolve by looking at a study. It resolves, or doesn't, by looking at what you're actually running. Right now, in the ordinary hours when no one is watching and you have forgotten to perform anything at all. That is the signal. That is what reaches the person across the table tonight. That is what the body next to yours in the dark is, quietly and without either of you choosing it, beginning to mirror. There is a woman in Sao Paulo. Her name has been kept out of the coverage at her request, but the story itself was reported in Folha de São Paulo in March, corroborated by the hospital staff who treated her and by the researcher who later interviewed her for a study on acute stress response in urban caregivers. She worked nights at a public hospital during the worst stretch of the respiratory crisis, came home each morning to an apartment that smelled of coffee she was always too tired to drink, and developed, over 14 months, what the cardiology team described as a resting heart rate and cortisol profile more consistent with someone in active emergency than with someone sleeping in her own bed. She was not sick. She was calibrated. Her nervous system had done exactly what nervous systems do. Read the environment it was living inside and tuned itself accordingly. The story made the paper because of what happened after. She left the hospital rotation, moved to a smaller city, did nothing dramatic, no retreat, no protocol, no intervention beyond ordinary quiet and ordinary sleep. And 6 months later, her markers had shifted measurably, not to some other person's baseline, to a wider range of her own. That story is the ground this chapter stands on, because the question her case raises is not whether stress leaves a biological trace. It does. The question is what that trace is and what you misunderstand about it when you treat it as damage. There is a particular kind of morning light that arrives sideways through glass, pale and uncommitted, the kind that doesn't yet know if it will become a real day. You reach for something warm without thinking, coffee, tea, whatever is near, and the heat moves up through the cup into the palms before you've decided anything about the day. The body is already orienting, already reading. That reading doesn't stop when the moment passes. It threads forward into every decision you make before noon, and you feel it not as thought, but as the particular texture of how available you are. That texture is what the woman in Sao Paulo recovered. Not a different nervous system, a different available range inside the one she had. The science underneath this is not metaphor. The law of vibration, the principle that every system broadcasts and receives according to its internal state, that what the organism emits shapes what it draws toward itself and what it registers as possible, is not contradicted by biology. It is expressed through it. The glucocorticoid receptor gene and the methylation patterns that determine how sensitively it fires is one of the mechanisms by which vibrational state becomes embodied. The organism attunes, and what it attunes to consistently, over time, rewrites the physical substrate of its own receptivity. The law of attraction operates in this same territory, not as wish, but as resonance. A nervous system calibrated to threat does not simply feel afraid. It selects for threat-confirming information, filters toward evidence of scarcity, orients toward outcomes consistent with the frequency it is already broadcasting. This is not mysticism. It is a description of how a tuned instrument works. The instrument does not attract randomly. It amplifies what it is already set to receive. which is why the woman's story matters past the cardiology. She did not reprogram herself through effort. She changed the inputs, allowed the system's inherent plasticity to respond, and the tuning shifted. The law of vibration says, change the state and the signal changes. The body said the same thing in numbers. And here is where the distinction arrives. The one that determines whether any of this is useful to you on an ordinary Tuesday, or whether it remains a theory you carry without ever feeling. There are two ways to bring a practice to your own history. They can look identical from outside. They feel nothing alike from inside. The first way is campaign. You bring the morning breathing, or the stillness, or the cold water, or the walk against something. Against the anxiety, against the inheritance, against the version of yourself that formed under conditions you didn't choose. The practice is aimed, and aimed practices tighten the instrument. Not metaphorically. Research from the Salk Institute examining chromatin remodeling in adult neural tissue found that the methylation changes associated with durable expression shift were more pronounced in low arousal, sustained attention states than in high effort, high urgency ones. The body rewrites itself more readily when it is not braced. A system fighting its own history is physiologically still fighting. The cortisol profile doesn't know the enemy changed. The second way is occupation. Not correction, presence. You're not trying to become someone else. You're creating the internal conditions under which a wider range of what you already are can surface. The difference is felt as texture. Campaign practices carry a held breath beneath the breath, faint, persistent, exactly like the sound a building makes when the wind is very slightly wrong. Occupation feels more like what happens when you finally set that cup down on the table and let the warmth do what warmth does without being asked. The woman in Sao Paulo didn't fight. She stopped fighting, and the system, finding less urgency to maintain, found a different register. Here is what must be said plainly, because the channel this belongs to does not traffic in comfortable half-truths. The inheritance is real. The calibration laid down across the early years of a life, by environment, by the bodies of people who raised you, by the epigenetic signatures of stress that transmit across generations not as malice, but as preparation. That calibration runs underneath conscious intention the way a foundation runs underneath rooms. You don't see it. You feel it as the speed at which concern becomes catastrophe, as the particular restlessness that has no source you can name, as the way certain silences feel like the edge of something dangerous. That is not damage. That is the system having paid attention. The organism was trying to survive. What was written into you was, in its own imperfect and sometimes painful way, a form of care, a biological attempt to send you into the world the previous body knew, prepared. You were not broken. You were informed. And that distinction, informed rather than broken, changes the vibrational state you bring to every practice, because the frequency of self-correction and the frequency of self-understanding are not the same signal. They do not draw the same things toward you. They do not remodel the same tissue. The law of attraction is precise about this, and the biology agrees. What you emit in the act of practicing determines, to a measurable degree, what the practice can return. The inheritance doesn't disappear. It becomes one voice in a larger room. The work, ordinary, unhurried, accumulated across unremarkable mornings, is the patient addition of other voices. not to silence the old one, to give it company, and in that company, less authority over the whole. The light through the window is still doing what it does. The cup is still warm if you let it be. The organism is already listening. The only question is what frequency you are broadcasting when you meet it. The question that was left open a few chapters back deserves an answer now. Reprogramming, we said, is not erasure. It doesn't ask you to pretend the inheritance didn't happen, to scrub the early years clean, to act as though the epigenetic markings written into your cells by someone else's hard life were never there. So what actually changes? Not the blueprint, the relationship to it. And that distinction, quiet as it sounds, is the whole thing. There's a study out of Stanford that most people in the wellness world have half heard of and almost entirely misunderstood. Ali Akram's work on mindset and physiological response. Not mindset in the motivational poster sense. Mindset in the measurable cellular sense. In one of her earlier experiments, she took hotel cleaning staff, people who spent their days in physical labor, and divided them into two groups. One group was told, with data, that their daily work already met the Surgeon General's recommendations for exercise. The other group received no such information. Same work, same bodies, same hours, different information held internally. 8 weeks later, the group that had been told, that had absorbed the information and changed their frame, showed measurable differences. Lower blood pressure, reduced body mass index, different waist to hip ratios. The work hadn't changed. The body's interpretation of the work had. Crumb's lab has gone on to examine how mindset shapes cortisol response, how the framing of a stressor as threat or as challenge changes not just mood, but actual hormonal output in the moment. The ghrelin response, the neuroendocrine cascade. The body is not just executing the protocol. It is reading the intention behind it, which brings us to something specific. Richard is 61. He found breathwork 2 years ago in the aftermath of a health scare, the kind that reorganizes a life without asking permission. His cardiologist had mentioned stress management. His adult son sent him a link. He started twice a day for 7-8 pattern, mornings and before bed, in the chair by the radiator in his bedroom, where the carpet is slightly worn through from years of the same footpath between door and window. He did this faithfully for 6 months. Blood pressure came down slightly. Sleep improved, but something still felt mechanical about it. He told a friend, "It feels like taking medicine. I do it because I'm scared not to." That is an important sentence. Not because it's wrong, because it's honest. And because the physiology of what happens when you breathe deeply from fear and what happens when you breathe deeply from a different internal position are not identical. The vagal tone shift is real in both cases. The parasympathetic system engages either way. But the secondary neuroendocrine environment, what the body is swimming in before and after, differs. Crumb's framework suggests the body doesn't only measure the action. It measures the context the action is embedded in. A breath taken as a desperate negotiation with mortality arrives in different tissue than a breath taken from a place of relative safety. Not wildly differently, not a dramatic reversal, but measurably differently. The difference between a system receiving the breath as relief and a system receiving it as correction. This is where the law of vibration earns its place in the conversation, and not as metaphor. Every cell in the body operates at a measurable electromagnetic frequency. That frequency shifts with physiological state. A nervous system held in chronic threat produces a different vibratory signature than one that is found, even briefly, something closer to coherence. The law of attraction, which travels with this everywhere it goes, is not about wishing. It is about signal, what you emit from that internal environment. Hormonally, neurologically, electromagnetically, is what your system is primed to recognize, repeat, and draw forward. The body under sustained threat keeps finding evidence that threat is the appropriate response. Not because life is conspiring against it, because that is the frequency it has been tuned to receive. Change the frequency, and the same external circumstances begin producing different downstream chemistry. Same inputs, different resonance, different yield. Richard's shift wasn't dramatic. He didn't restructure his mornings around a system. He simply started doing the breathing earlier, before the phone, before anything that asked something of him. Same pattern, same duration, but preceded by a few minutes of sitting at the kitchen table with both hands around his mug, the ceramic warm, the smell of the coffee rising in the still air. The neighbor's dog not yet started in on its morning complaints. Nothing resolved, nothing scheduled, just the weight of the mug in his palms and the kitchen holding its quiet. The breathing that followed wasn't different in form, but the body it landed in was in a different state. He wasn't reaching for a tool to fend something off. The frame had shifted, not because he constructed one, but because the 2 minutes before had already moved him somewhere else. His blood pressure readings across the following year, confirmed across 3 separate visits, were the best in 4 years. That isn't proof of anything singular. Too many variables. But it rhymes precisely with what Crumb's controlled work has found for over a decade. That the internal context surrounding a behavior is not decorative. It is metabolically present inside the behavior itself. The milkshake study makes this almost uncomfortably clear. The same shake, consumed with a label reading indulgent versus sensible, produced different ghrelin responses. The molecules were identical. The frame was not, and the body followed the frame. For someone in their 50s who has spent decades doing things because they were afraid of what would happen if they didn't, this is not a small finding. The supplement taken in quiet dread. The walk done to outrun the diagnosis. The sleep tracked anxiously, lying awake wondering whether the number will be enough. All of it is real effort, real discipline. It should count, but the body is reading the emotional signature wrapped around every one of those actions, and that signature influences what the action produces. A nervous system in threat mode performing a health behavior is doing something genuinely different at the level of downstream chemistry than a nervous system in relative coherence performing the exact same one. First is repair under fire. Second is something else, maintenance from a different ground. Same action, different internal environment, different result. This is what changes for someone who has followed this material across its full arc. Through the inherited biology, the epigenome, the slow realization that ancestors arrive silently inside their descendants and keep showing up. What changes is not the to-do list. The breathing is still breathing. The walks are still walks. The sleep is still sleep. What changes is the frequency those actions are taken from. The vibratory state the body is in when the behavior begins. And according to both Crumb's empirical framework and the mechanics of the law of vibration, that starting frequency is not incidental. It is, in measurable terms, the primary variable. The question that follows is the practical one. How does any of this survive contact with an actual morning? The alarm sounds. The phone is already lit. The day has its own gravitational pull, and none of it has been paying attention to this. There's something that happens in the body in the minutes immediately after waking, a specific neurological condition that exists briefly before the day colonizes it. And what you do or don't do inside that window shapes the frequency everything else happens from. What that window looks like, how narrow it is, and why most people have been spinning it in exactly the wrong way is where this goes next. The question that was left open a few chapters ago, whether intention changes the physiology of a practice, not just the outcome, lands here. It does. That is not a motivational claim. It is what the data keeps returning to from different angles in different labs with different populations. The internal condition of the person doing the thing changes what the thing does to the body. The breath taken with indifference is not the same breath taken with presence. The walk taken under obligation is not the same walk taken with even a thin thread of genuine choice. The molecule absorbed in dread is not the molecule absorbed in calm. The biology does not treat these as equivalent inputs. It never did, which is exactly why what you do with this conversation matters as much as the conversation itself. Not the information, the conversation. There is a difference. Information sits on a shelf. A conversation changes the person who is in it. That is what this channel is trying to be. Not a feed, not a digest. A place where the news comes in fully, honestly, without softening the weight of it, and then shifts slightly in your hands while you're holding it. Not because the facts change, because the question underneath the facts changes. Here is what the last hour has been pointing toward. If you can feel it now rather than just understand it, you're not entirely downstream of your biology. The systems in your body received signals from environments that existed before you could name a single thing in the world, before language, before memory, before any capacity to choose. That part is true, and it needed to be said plainly. But the signal does not stop arriving. The system that learned to respond to threat is still taking in information. Right now, that is not a comfort. It is a mechanism, and mechanisms can be worked with if you know what you are working with. This is where researchers like Bruce McEwen spent decades, not on dramatic interventions, but on how ordinary, sustained shifts in environmental load change the regulatory behavior of stress systems over time. Not supplements, not clinical programs, just a consistent reduction in the kind of low-grade, chronic activation that most people have stopped noticing because it has become the baseline. McEwen's allostatic load work showed that the body keeps a running account of that activation, and that the account can be drawn down as well as built up. The direction is not fixed. The body was listening the whole time. It is listening now. This channel exists to keep being a different kind of input. Each video starts from something real, a policy decision, a study, a geopolitical shift, something you would encounter scrolling or on the radio, and works it the way it deserves to be worked, with the weight it carries, without pretending the world is simpler than it is. And then it asks the question underneath the event. Not what happened, what activates? what it means to hold a real and difficult thing from a slightly different internal position. Not denial, not managed distance, but a different posture. Because the posture is not irrelevant. You know that now. This video made sure of it. Subscribing here is not complicated. There is nothing being sold. The next video will come from a real event, a real finding, a real tension, and it will follow the same thread this one has been pulling. The only variable is whether you are in a room when it does. Some pieces of a conversation land differently depending on when they reach you. The same material at the wrong moment does less. That is not a sales line. It is just how sequence works in an argument that is still building. You have been here for the better part of an hour. What was happening in you while you watched was not just comprehension. The part of your nervous system that processes threat and safety was taking cues from the tone, the pacing, the absence of manufactured urgency. That is not nothing. It is, in fact, the whole point that the medium is doing something to the receiver, and that what it does depends on what kind of signal it is carrying. This one was not trying to alarm you. It was not trying to reassure you either, which would have been dishonest given the subject matter. It was trying to be accurate about what the research actually shows, about what the world is actually doing, about what you might actually be able to do with that, given a clear picture of the machinery involved. The final chapter closes the argument. One thing was left unresolved deliberately, and it gets resolved there. Not dramatically, just completely. Be there for it. The question that opened this story. If something in you was configured before you even existed, what does that change about who you are now and what you are configuring next? That question was never rhetorical. It was a compass, and this is where it points. Go back to Uppsala for a moment. Not to the data, not to the methodology, not to the numbers scrolling across the researcher's screen in that spare Scandinavian office where the radiator ticks quietly and the morning light comes in flat and gray off the snow. Go back to the man sitting in front of those numbers. Because something happened to him during the years of this research that the paper itself does not record. He started the project looking for damage. That is the honest framing of where epigenetic inheritance research begins. It begins with the assumption that what gets passed down is burden, famine signatures, stress imprints, trauma encoded in the regulatory layer above the genome. The early literature reads like an inventory of wounds, and so the researchers arrive at their data expecting to find a ledger of what went wrong. What the Uppsala group found, after years of tracking how early life conditions, including conditions that preceded birth, conditions experienced by the mother during gestation, conditions absorbed before any conscious moment of the subject's life, what they found was not a ledger of wounds. It was a record of adaptation. The body had not simply recorded suffering. It had responded to it, reorganized around it, built compensations, sensitivities, altered thresholds, not randomly, but in patterns that made a kind of biological sense when you understood the environment they were anticipating. That is where the story turns. Because the language of epigenetics, up to a point in the literature, had been the language of determinism dressed in scientific clothing. You carry what was done to you. You express what your ancestors survived. You are at the cellular level, the echo of things that happened to people who are no longer here. And if you absorb that framing, and a lot of people did because it came wrapped in peer-reviewed authority, it left you with a particular feeling. Not quite helplessness, something more specific. The feeling of arriving late to a game whose rules were written without you. That feeling was understandable. It was also, the Uppsala data suggests, incomplete. Here is what the data actually shows when you read it without that framing. The epigenetic layer is not a sealed record. It is a living interface. It responds to environment, not just the environment of the womb, not just the environment of early childhood, but the environment of right now. The methyl groups that sit on the regulatory regions of your DNA and determine which genes speak loudly and which ones speak quietly. Those are not installed once and locked. They shift. They respond to sustained signals. They are, in the most literal biological sense, listening. The researcher in Uppsala was not looking at a photograph of the past. He was watching a process that had no intention of stopping. That distinction between a record and a process is the thing this entire video has been building toward without announcing it. Because a record asks you to accept what it contains. A process asks you to consider what you are contributing. The science of what modulates the epigenome in adult life is not thin. Research from the Karolinska's adjacent institutions, from groups at UCL, from longitudinal cohort work at the Finnish Institute for Health and Welfare, tracking thousands of subjects across decades. This body of work converges on several signals that reliably shift epigenetic expression in adults over 45. Sustained aerobic effort, sleep architecture, specifically slow-wave depth, nutritional signaling, particularly around methyl donor compounds, folate, choline, the B vitamin family, and something that the literature handles more carefully but cannot avoid, the chronic emotional valence of a person's inner life. Not mood, not optimism as a personality trait. The sustained baseline frequency of how a nervous system operates day after day. That last signal is the one the popular conversation tends to either overstate into spirituality or dismiss into irrelevance. The actual data sits between those two extremes. What the Finnish cohort work shows, and what a 2021 paper in Psychoneuroendocrinology documented using DNA methylation arrays in a sample of adults between 48 and 72, is that individuals whose autonomic profiles, measured via heart rate variability and cortical arousal patterns, reflected sustained low-threat states, showed measurably different methylation signatures at specific gene promoters associated with inflammatory regulation compared to individuals whose autonomic profiles reflected sustained high vigilance states. The difference was not dramatic in any single measurement. Accumulated over years, was not trivial. The body is not recording your worst days. It is averaging your ordinary ones. That detail is worth sitting with before moving further, because it shifts the terrain of the conversation in a way the crisis-focused framing of most longevity content never quite reaches. The acute stressors, the bad diagnosis, the season of loss, the year that bent something in you, those matter, but they are not the primary signal. The background frequency is the hum of how you move through a Tuesday afternoon, the state you return to after the news, after the difficult call, after the thing that was supposed to ruin the week but didn't quite, the baseline you rebuild each time, or fail to rebuild, or rebuild slightly lower than before. That baseline is environmental, and the epigenome treats it as such. Which means the question of what frequency a biological system sustains when it is not under direct demand is not a metaphor borrowed from physics. It is a mechanism, That sustained frequency is the signal the regulatory layer above your genome is actually reading. Not what you feel in the peak moments, what you return to, what you live in between. The popular literature reaches for spiritual language here and loses the thread. The dismissive literature waves it away and loses a different thread. What the data shows is more specific than either. The nervous system's resting posture, held consistently across months and years, leaves a legible mark on which parts of a genome are being expressed and which are being quieted. That is the ground level claim. The next one is larger. The epigenetic marks that shift in your lifetime do not all reset between generations. The literature on transgenerational epigenetic inheritance in humans is still contested at the edges. The mechanisms are clearer in animal models than in human longitudinal data, but the convergence of evidence is sufficient that researchers who were skeptical a decade ago now speak with more care about what they dismiss. A 2022 paper in Nature Reviews Genetics, reviewing the human evidence specifically, concluded that while complete generational reset of epigenetic marks does occur from many loci, it is incomplete for others, and the patterns of what is retained versus erased remain poorly understood and likely context dependent. The researcher in Uppsala was sitting with something like that. Not resolution, not a tidy conclusion. The particular quality of attention that comes when a dataset has moved further than the original hypothesis and you're still catching up to where it went. He had started the project expecting to find evidence of inherited damage, evidence that the past pressed forward into biology with the weight of something fixed. What the data had done, slowly, across years of returned samples and adjusted models, was taken the concept of inheritance and made it less like a verdict and more like a current. Something that moved through people the way weather moves through a landscape, shaped by the terrain, shaping it in return, but belonging to neither. What was configured before you were born is real. The evidence for that is solid enough that dismissing it requires deliberate ignorance. But the framing that turns that fact into passivity, the framing that says you're the endpoint of that configuration, the final expression of something already closed, that framing is not what the data supports. The data supports something less comfortable and considerably more demanding. You're not the endpoint. You are a continuation. The process that began before your birth, in the lives of people you may have known briefly or not at all, in the conditions they navigated, in the baseline they sustained or failed to sustain, that process arrived in you, and it is still running. And what you do with the years you have left in this body, the baseline you build or fail to build, the sustained signal you contribute to the system, that becomes part of what continues. Not as legacy in the way the word usually gets used, the softened, retrospective version of it, as biology, as the actual epigenetic environment your body creates that may, through mechanisms still being mapped, reach forward in ways that exceed your individual lifespan. That is a different kind of responsibility than the longevity industry typically sells. The industry sells you a body that lasts longer. What the Uppsala data is gesturing toward is something more precise, a body that transmits differently. The light in that office has shifted by now. Later in the morning, the researcher has moved on to other files, other data, the ordinary forward motion of a workday. The radiator still ticking. Outside, the same flat Nordic light, but angled slightly the way it does when the morning has committed to being morning. a man at a desk, doing the unglamorous work of sitting with what the numbers actually say rather than what he came and expecting to find. The inheritance you carry is real. The capacity to alter what you carry forward is also real. Those two facts do not cancel each other. They create a specific kind of pressure, not the pressure of guilt, not the pressure of optimization, but something quieter and harder to put down. Not what was done to me before I arrived, but what am I right now for what comes next? That question does not resolve easily and is not meant to. It follows you out of the room and sits with you through the ordinary hours, the Tuesday afternoon, the background hum of a life in motion, asking in its quiet way what frequency you are sustaining and what you're passing on.